Okay, good morning all of you. Fine, so I hope you have understood everything in the last session. So we had started with your MCQs. We have finished till question number 101. Please open your books, go to the MCQ pages, and then you start seeing the lecture. We start further ahead. Question number 102. Emasculation is removing stamens before anthesis. So what is anthesis? Opening of flower bud is called as anthesis, and if you want to do hybridization, and so we want to make a bisexual plant become unisexual, then you will cut off the stamen or androecium before the flower opens, so that self-pollination does not take place. So what is that emasculation, sir? Removal of stamen before opening of the flower, called as anthesis. Question number 103. When phenotypic and genotypic ratio is the same, then it is an example of incomplete dominance or codominance. If you remember, in incomplete dominance and codominance, phenotypic and genotypic ratios are the same, 1:2:1. So what is 1:2:1 phenotypic and genotypic ratio an example of? Incomplete dominance or codominance. 104. When two genes control a single character and have a cumulative effect, the ratio is, I hope you remember, Nielsen L has conducted it in wheat plant, and red kernel, white kernel variety of wheat in the F2 generation gives you a ratio of 1:4:6:4:1. 1:4:6:4:1 when two genes control. If three genes control, then what is the ratio? 1:6:15:20:15:6:1 when three genes control. 105. Genes located on the same locus but showing more than two different phenotypes are called as pleiotropic genes. HBS with another HBS forms sickle cell anemia death. HBS with an HBA shows sickle cell carrier, which also shows anemia and oxygen deficiency, breathlessness. But it also offers resistance against malaria. So what are such genes called as? Pleiotropic genes. One gene controlling multiple phenotypes. Then 106. If a cross between two individuals produces offspring with 50% dominant character and 50% recessive character, then the genotypes of the parents are B. A capital small a into small a small a. What is this an example of? A test cross. And what will be a test cross ratio? 1:1. In other words, 50% dominant character, 50% recessive character. 107. Allelism refers to alternative forms of genes at a given locus. When you have two alleles, capital T and small t, these are alternative forms of genes found on the same locus of homologous chromosomes, then what are such genes called as? These are called as alleles. So they are alternative forms of a gene at a given locus. 108. Mendelian principles are not applicable in case of B. Asexually reproducing forms. अगर asexual reproduction हो रहा है तो meiosis will not take place, and if meiosis does not take place, then law of segregation and purity of gametes will not take place, law of independent assortment will not take place. So meiosis is necessary for Mendelian principles. Asexual reproduction में Mendelian principles are not holding true. Then 109. The genotypic and phenotypic ratio in a monohybrid cross remains the same in case of intermediate inheritance, which is also called as incomplete dominance. So intermediate inheritance also called as incomplete dominance. 110. Mendel was lucky in discovering the principles of inheritance as B. He did not encounter linkage between the genes for the characters he considered. Mendel was lucky, he studied 7 different contrasting characters and all 7 were found on different chromosomes. If they were found on same chromosome, then they would have not shown independent assortment. So Mendel was lucky, he didn't encounter linkage, otherwise independent assortment nahi hota tha. 111. 4 children belong to the same parents having the following 4 blood groups A, B, AB and O. Hence the genotypes of both parents are. Nice question. Answer C. One parent is heterozygous for A and another parent is heterozygous for B. So समझो blood group is I capital A small I and I capital B small I. So these are 2 different blood groups and समझो इनके बीच में meeting हुआ, then what kind of inheritance patterns are possible? So if these 2 are the patterns, then the child may get blood group IA, IB as parents. Then that would form IA, I small I, IB small I, and that would be blood group A and blood group B. But समझो the child gets this pattern and this pattern. Then sir, what will be again the blood group? I capital A small I capital B small I, and the blood group will be A and B. And समझो the child gets this pattern and this pattern. Then that means it is I capital A, I capital B and small I. Then what would be the blood group AB and O? So इधर कौन सा blood group है A, इधर कौन सा blood group है B, इधर कौन सा blood group है AB, codominant, और इधर कौन सा blood group है O. So when will a child have A group, B group, AD group, O group? When both parents are heterozygous for A group or heterozygous for B group. So a parent heterozygous for A group and B group can have all types of children, A group, B group, AB group and O group. Nice question, mark as important. 112. In a typical Mendelian cross, which is a dihybrid cross, one parent is homozygous for both dominant traits and another parent is homozygous for both recessive traits. In the F2 generation, both parental combination and new recombination appear. The phenotypic ratio of parental combination to new recombination is 10:6. We had done this last time. Yellow round, green wrinkled. In the F2 generation forms 9 yellow round, 3 yellow wrinkled, 3 green round, 1 green wrinkled. So how many are parental combination? Yellow round are 9, green wrinkled is 1. So yellow round 9, green wrinkled 1. So 10 are parental phenotypes and 3 are yellow wrinkled, 3 are green round. So 3 plus 3, 6 are new phenotypes. So what is the ratio of parental to new phenotype? 10:6. Then we have 113. In a Mendelian dihybrid cross, when heterozygous round yellow or fence cross, round green offspring will be represented by what genotype? Heterozygous round yellow, say green and the round seeds कौन से मिलेंगे? तो सर ये है green round और ये है green round. So which are the green rounds? You get small y and this will be small y, capital R will be green and round, and small y, capital R will be green and brown. So what is your option? D. D में you have capital R, small y, and capital R, small y. These will become green and round. So answer D, capital R, small y, and capital R, small y are green and brown. And then we have 114. Inheritance of flower color is an example of incomplete dominance seen in Antirrhinum major, which is dot flower. Then 115, the graphical representation to calculate the probability of all genotypes of an offspring in a genetic square is called as a Punnett square. What is that called as a Punnett square? 116, the genotype of a plant showing dominant phenotype can be determined by a test cross. एक टेस्ट क्रॉस से मालूम पड़ेगा कि whether it is homozygous dominant or whether it is heterozygous dominant. 117, which of the following cannot be explained on the basis of Mendel's law of dominance? Mendel's law of dominance, the discrete unit controlling a particular character is called as a factor, is proved by Mendel's law of dominance. Out of the pair of factors, one is dominant, other is recessive, is again a law of dominance. B, factors occur in pairs, is again expressed by law of dominance. But C, alleles do not show blending and both characters recover in the F2 generation is not law of dominance, it is law of segregation and purity of gametes. So C is your answer. Alleles do not show any blending and both characters recover in the F2 generation is not based on law of dominance but law of segregation and purity of gametes. Then 118, select the correct statements from the ones given below with respect to a dihybrid cross. Option D, tightly linked genes on the same chromosome show very few recombination. अगर एक ही क्रोमोसोम पे दोनों जीन्स है तो dihybrid cross होगा. What will happen during formation of gametes, sir? The tightly linked genes, say capital T, capital R, will go in one chromosome. The other alleles, small t, small r, go with another chromosome, and independent assortment will not take place. अगर independent assortment नहीं हुआ, then what will be the plant producer? Capital T, capital R से tall and round बनेगा. Small t, small r से dwarf and green बनेगा. In other words, independent assortment will not take place and you will get parental combination. So new recombination का possible है only when independent assortment takes place. And when is independent assortment possible? When genes are found on separate chromosomes. Then independent assortment takes place. If genes are found on same chromosomes, then independent assortment will not take place. So nice question this one. What will happen if there is a dihybrid cross? Tightly linked genes on the same chromosome show very few recombination and mainly show parental combination. Then 119, the F2 genotypic ratio of a monohybrid cross is 1:2:1. Phenotypic ratio 3:1, genotypic ratio 1:2:1. 120, if a heterozygous tall plant is crossed with a homozygous dwarf plant, then the percentage of progeny having dwarf character is 50%. A heterozygous tall plant is crossed with a homozygous dwarf plant, then this is an example of a test cross. And what will we get over here? Capital T, small t, small t, small t, capital T, small t, small t, small t. 50% will be tall, 50% will be dwarf. So heterozygous tall plant crossed with homozygous dwarf plant, percentage of progeny having dwarf character is 50%. 121, to determine heterozygosity of a cross, one has to perform a test cross with the recessive parent. 122, test cross is a cross between a hybrid and a recessive parent. That's C. A hybrid and a recessive parent is a test cross. 123, marked important, a gene that masks another non-allelic gene's expression is called as epistatic. Something new for you. If you have capital T, small t, capital R, small r, what will capital T become? Dominant over small t. What will capital R become? Dominant over small r. And that is called law of dominance. Now, in certain plants, अगर capital T small t, capital R small r है, capital T will become dominant over small t, but capital T will become dominant over capital R also. So, plant will be tall, but उसमें round seeds नहीं आएंगे, उसमें wrinkled seed ही आएगा. क्योंकि not only capital T is dominant over small t, capital T is dominant over capital R. So, what is that phenomenon called as? It is called as epistasis, where capital T becomes dominant over capital R. It is called as epistasis, and such genes are called as epistatic genes. And the gene which gets suppressed is called as hypostasis. So capital T becomes dominant over capital R. What does capital T become? Epistatic. What does capital R become? Hypostatic. So capital T is epistatic, capital R is hypostatic. Nice question. ये याद रखना. Then 124. Some individuals with blood group A may inherit genes for blonde hair, while other individuals with blood group A may inherit gene for brown hair. This is best explained by principle of independent assortment. If somebody has got chromosome with I capital A and somebody has got I capital B, and then there is another chromosome with B for blonde hair and another one with B for brown hair. Or rather, this we will say small b for blonde hair, which is recessive, capital B for brown hair. So, sir, what can it lead to? It can lead to A group with blonde hair, or it can lead to A group with brown hair. So, sir, ये भी हो सकता है, ये भी हो सकता है. So, what is this phenomenon called as independent assortment? So, this is independent assortment taking place. Then, when two hybrids, smaller, capital T, small t, and capital R, small t are crossed, what will be the phenotypic ratio of the offspring? Nice question. Suppose it is smaller, capital T, small t into capital R, small t. So we just change it here and change it here. तो यहाँ कैपिटल आर स्मॉल आर हम लोग यहाँ ले लेंगे कैपिटल आर स्मॉल आर कैपिटल टी स्मॉल टी इनटू and we have taken small r here, so small r small t. So what does this become an example of a dihybrid test cross? And last lecture हमने किया था what is a dihybrid test cross ratio? 4:4:4:4. In other words, 1:1:1:1. So what will be your answer over here? 1:1:1:1. Then 126, genes exhibiting multiple effects are known as pleiotropic genes. One gene produces multiple phenotypes. What is that called as? Pleiotropic genes. Then 127, in case of incomplete dominance, what will be the phenotypic ratio of the F2 generation? 1:2:1. 128, genes are made up of polynucleotides. Genes are of course made up of multiple nucleotides. About 3000 nucleotides का एक gene होता है, so it is a polynucleotide. 129, a dihybrid for a qualitative trait is crossed with a homozygous recessive individual of its type. The phenotypic ratio is a dihybrid, capital R, capital D, small t, is crossed with a homozygous recessive individual. Homozygous recessive, small r, small t. What is this an example of a dihybrid test cross? And what is the dihybrid test cross ratio? 1:1:1:1. 130, nice question. improvement of human race through hereditary qualities is called as eugenics. अगर हम अच्छे genetic changes human race में डालते हैं, useful mutations are passed on in the human generation and it spreads in the human generation, then that means human race will improve. And what is improvement of the human race by genetic introduction called as eugenics? ये याद 131, 132 we don't have it in the syllabus. Please cancel that. 131, 132 you don't have it. Please cancel that. 133, beautiful question. In garden P, yellow color of cotyledon is dominant over green and round shape. Garden P में yellow color is dominant over green and round shape is dominant over wrinkled. Then check this question here. Beautiful question. Yellow dominant over green, round dominant over wrinkled. When a pea plant with yellow and round seeds is crossed with a plant having yellow and wrinkled seeds, the progeny shows segregation of all four characters. Underline that. It shows segregation of all four characters. Then the probability of obtaining green round seeds in the progeny of this process. Underline the word segregation of all four characters. तो F2 जनरेशन में क्या आना चाहिए? चारों चार कैरेक्टर आने चाहिए. येलो राउंड भी आना चाहिए, येलो रिंकल्ड भी आना चाहिए, ग्रीन राउंड भी आना चाहिए, ग्रीन रिंकल्ड भी आना चाहिए. So what will happen if the plant is yellow round and yellow wrinkled? Yellow round will be capital Y, small y, capital R, small r, and yellow wrinkled will be capital Y, small r, small r. तो ये है yellow round, ये है green wrinkled. Now our logic is to get all four characters: yellow round, yellow wrinkled, green round, green wrinkled. तो इसमें से gametes कौन से बनेंगे? Capital Y, capital R. And then we will have capital Y, small r, then we will have small y, capital R, and then we will have small y, small r. तो ये gametes बन गए. और इसमें से कौन से gametes बनेंगे? Capital Y, small r, and you will get small y, small r. Now, what will this become? Capital Y, capital R, small r. What will this become? Capital Y, small r, small r. What will this become? Capital Y, small r, small r. What will this become? Capital Y, small r, small r. What will this become? Capital Y, small r, small r. What will this become? Capital Y, small r, small r. What will this become? Small y, capital R, small r. What will this become? Small y, small r, small r. Now check this out. What will this plant become? Yellow and round. What will this become? Yellow and wrinkled. What will this become? Yellow and round. What will this become? Yellow and wrinkled. What will this become? Yellow and round. What will this become? Yellow and wrinkled. तो अब तक हमको आया येलो राउंड और ये आया येलो रिंकल्ड एंड व्हाट विल दिस बिकम? ग्रीन, small y and round. So this will become green and round, and what will this become? Green and wrinkled. तो हमारे पास कौन-कौन से प्लांट्स हैं? We got yellow round also, we got yellow wrinkled also, we got green round also, and we got green wrinkled also. So sir, we are having yellow round, yellow wrinkled, green round, green wrinkled. We got segregation of all four characters. So that is the data. what is the probability of obtaining green round seed in this progeny? तो green round seed का probability कितना है? 1 out of 8 plants is green and round. 1 out of 8 plants is green and round. So what's your answer? 1 upon 8. 1 upon 8 will be green and round. 1 upon 8 will be green and round. So cross हमने कौन सा लिया? Yellow round, yellow wrinkled. We took capital Y, small y, capital R, small r, capital Y, small r. And इससे yellow round, yellow wrinkled, green round, green wrinkled. We got segregation of all 4 characters. And how many we got green and round? We got 1 plant. We got green and round. So 1 out of 8 is green and round. Mark that important. 134. A true breeding plant produces red flowers, is crossed with a pure plant producing white flowers. Allele for red color of flower is dominant. After selfing of the plant of the first filial generation, proportion of plants producing white flowers in the progeny would be 1/4. So this is a monohybrid cross. If we have capital R or capital R crossed with small r, what will we get in the F1 generation? Capital R, small r. And what will we get in the F2 generation? Capital R, small r and capital R, small r. So capital R एक मिला, capital R, small r एक मिला, capital small r एक मिला, small r एक मिला. So sir, how many white flowers we got? 1 white flower out of 4. 3 will be red flower, 1 will be white flower. So 1/4. 135. A character which is expressed in a hybrid is called as a dominant character. Recessive will express only in homozygous condition. In a hybrid, who will express? In a hybrid, only the dominant character will express. 136. The gene which controls many characters is called as the pleiotropic gene. One gene controlling multiple phenotypic characters is called as a pleiotropic gene. 137. The phenotypic ratio in the F2 generation of a dihybrid cross is 9:3:3:1. 138. Lack of independent assortment of 2 genes A capital and B capital in fruit fly is due to linkage. If A and B are found on the same chromosome, means they are linked together, then they will not separate during meiosis. In other words, independent assortment will not take place. So when are 2 genes never showing multiple assortment or independent assortment? 2 genes will not show independent assortment if they are linked together on the same chromosome. So what is that phenomenon called as linkage? Then 139, very nice question, marked important. Grain color in wheat is determined by 3 pairs of polygenes following the cross A capital, B capital, B capital, C capital, C capital, dark color into small a, small b, small c, light color. What proportion of the progeny is likely to resemble either parent in the F2 generation? So sir, this is a nice example of a trihybrid. So sir, ये हमने किया था in case of polygenes. Polygenes में what will you have if we have A capital, B capital, C capital, small a, small b. Intermediate will become A capital, small a, B capital, small b, C capital, small b. And F2 generation mein we got one plant which is A capital, B capital, C capital, and one plant which is small a, small b, small c. So out of 64, 2 plants are showing the parental genotypes. Out of 64, 2 plants are showing parental genotype. So what percentage of progeny is likely to resemble either of the parent? 2 out of 64, which is 1 out of 31. 32, which is less than 5%. So which is less than 5%. So nice question. What is the proportion of progeny likely to resemble either parent in F2 generation? Less than 5%. 140. Mating of an organism to a double recessive in order to determine whether it is homozygous or heterozygous for a character under consideration is called as a test cross. It is a dihybrid test cross. What is that called as a dihybrid test cross? 141. In short-horned cattle, genes for red capital R and white small r coat color occur. A cross between red capital R and white small r produced Rowan coat capital R small r. This is an example of codominance. A red and a white forming a patches. Rowan is a codominance. Which of the following genes show heterozygous character? A. Capital R small r is showing heterozygous character. 143. Which of the following is best suited for codominance when both genes are dominant? So I capital A, I capital B दोनों dominant हैं. So उनका blood group क्या आएगा? AB. And what is that an example of? Codominance. So codominance कब होगा? When both genes are dominant, it will show codominance. 144. Genes when close together on a chromosome is known as linkage. And what will linked genes show? No independent assortment. Linked genes में independent assortment नहीं होगा. Then incomplete dominance is shown by mirabilis. Mirabilis, that is 4 o'clock plant, and snapdragon, dog flower, antirrhinum majus show incomplete dominance. 146. A condition where a certain gene is present in a single copy in a diploid cell is called as hemizygous. So sir, समझो एक cell के अंदर chromosomes are in a pair, but if one chromosome is single, it doesn't have another copy, तो उसका और एक chromosome नहीं है, then that condition is called as hemizygous. That condition is called hemizygous. So you have homozygous, you have heterozygous. When you have two chromosomes. If you have only one chromosome, then what is that condition called as hemizygous? Then 147, a dihybrid test cross yielding the result of 1:1:1:1 ratio is indicative of 4 different types of gametes produced in the F1 dihybrid. So sir, F1 dihybrid में क्या बनेगा? If it is F1 dihybrid, how many types of gametes are produced? 4. This is dihybrid, it will form 4 types of gametes and you will get a ratio of 1:1:1:1. So that is a dihybrid test cross. 148, if A capital, small b, and small a, capital B are crossed, then the phenotypic ratio of its F2 progeny, उधर F2 लिख दो, of its F2 progeny will be, yes, what is A capital, B small into small a, capital B. इसके अंदर वापस से just exchange the genes. So sir, if we do exchange of genes, this will become A capital, B capital into small a, small b. What will F1 generation show? A capital, small a, B capital, small a. And what will F2 generation show? A ratio of 9:3:3:1. So this is an example of a dihybrid cross. 149. When a dihybrid cross is fit into a Punnett square with 16 boxes. When a dihybrid is crossed into a Punnett square with 16 boxes, how many different types of phenotypes are possible? तो अगर हमने 16 boxes में dihybrid को fit किया है, तो कितने phenotype possible है? 4 phenotype. Get dominant 9, dominant recessive 3, recessive dominant 3, recessive 1. So how many maximum number of phenotypes are possible? 4. And 150, in a heterozygous condition, the individual expression of both alleles in the phenotype is exemplified by AB blood group. Agar AB blood group hai, then both alleles will express, and what is that phenomenon called as codon. So AB blood group, what is that phenomenon called as codon? Yeah. So we'll just take a small break over here and we'll come back and finish the rest of the question. Take care.